Please join me to welcome our second finalist, Dr. Xiaoqi Feng from the John Innes Center in the UK. And she's going to tell us something interesting about the epigenetic regulation in plants. So Professor Feng, please.
Can you see my screen?
Yes.
Right. Okay, great. I'll just change this to a laser pointer. Okay. Yep. Great, thank you. First, I'd like to thank the committee and Molecular Plant for this opportunity. And it is a great pleasure for me to participate in this very exciting symposium. So my lab's research has been really interested in the germline because these are the cells that bridge the generations and allow life to perpetuate. And we're particularly interested in a key aspect of germline development, which is this large-scale reconfiguration of chromatin that occurs in the germ cells. So the modifications on DNA and chromatin that do not change the underlying genetic code, such as DNA methylation and modifications on histones and usage of different histone variants, are collectively called epigenetic modifications. And to date, these epigenetic modifications have been well demonstrated to play essential roles in regulating genome functions. And the hallmark of germ cell development is the large-scale reprogramming of these epigenetic modifications. And defects in such reprogramming cause sterility in animals and plants, demonstrating that this process is essential for germline functions. So a central goal of my lab is to understand how epigenetic reprogramming regulates germlines. And conversely, I believe germ cells also act as models to understand general epigenetic mechanisms. And this is because the natural changes the germ cells experience expose aspects of the epigenetic pathways that are not obvious in more static systems. So the model my lab developed to study germ cell epigenetics is the male germline of Arabidopsis. So here, this cartoon shows that the male germline initiates as diploid meiocytes, which undergo meiosis to generate haploid spores called microspores. And then each spore divides once mitotically to give rise to a small generative cell and a large companion called the vegetative cell. And then this small red cell enters another round of mitosis to give rise to the two sperm cells in the pollen. And this entire developmental process is characterized by extensive chromatin reconfiguration and reprogramming. So studies, including those from my lab, have shown that there are distinctive reprogramming events following every step of this development and regulating different aspects of germline functions. And the great advantage of the system is that it is highly tractable. My lab developed techniques for the isolation of all these germ cell types that have one cell division apart. And to complement this, we developed live imaging tools and markers to look at these cells, and also protocols to perform epigenomic analysis using limited amount of input cells, including single cells. And another great advantage of the system is that Arabidopsis shares highly conserved chromatin pathways with crops, but it is far more tolerant to epigenetic mutations. So mutants that are typically lethal or sterile in crops are relatively happy and sterile and fertile in Arabidopsis, and this permits powerful genetics. So in other words, Arabidopsis is great for figuring out the mechanism, which then can be readily applied to crops. So using the tools we set up, my lab is studying the reprogramming of DNA methylation and histone modifications and histone variants, which are interconnected, and looking at how the environment modulates germline epigenetics. So today, I will mostly focus on the first two themes and tell you a few recent stories from the lab regarding DNA methylation and chromatin reprogramming. And I will also just very briefly mention our work on theme 3, the environmental modulation. Okay, so now let's first take a look at the DNA methylation in the germline. So in eukaryotes, DNA methylation mostly occurs on cytosines via the addition of a methyl group to the 5th carbon, called 5-methylcytosine. This is the most well-studied epigenetic mark to date, with essential roles in regulating the activities of genes and transposons in the genome. And in plants, there has been a long-standing, large amount of literature demonstrating stable transmission of methylation patterns across generations. And this has led to a long-standing question in plant epigenetics regarding whether methylation reprogramming occurs at all in the plant germlines. And a graduate student, James Walker, and the former postdoc, Hongbo Gao, they found that there is indeed methylation reprogramming. And this manifests in the distinctive methylation of hundreds of genes across the entire male germline. So you see this from meiocytes all the way to the sperm. And this methylation is absent from somatic cells. And we further discovered that this germline-specific methylation at genes suppresses gene expression in the meiocytes and regulates the splicing of a gene essential for meiosis. So this work established a new paradigm in which a cell lineage-specific methylation signature controls gene expression and cellular function. And recently, we have gone further into understanding the molecular mechanism driving this methylation reprogramming. And we found that the gene methylation in the male germline is induced by sRNAs transcribed from transposons with similar sequences. And this interestingly occurs in the neighboring nerve cells called the tapetum. So these tapetum cells, they can act in trance at both cellular and locus levels to target DNA methylation. So this work demonstrated the essential role of tapetum sRNAs in configuring the paternal epigenome to be inherited by the next generation. So besides this complicated molecular mechanism, we also have some exciting data that suggests that this pathway is modulated by the environment. So our data suggests that these tapetum sRNAs that drive germline methylation reprogramming are attenuated at high temperature. And this leads to the reduced methylation and activation of a heat response gene in the meiocytes. So now we are investigating the hypothesis that environmentally malleable epigenetic information flows from the soma to the germline and mediates germline stress response. And besides Arabidopsis, we have also extended our study to other species to understand the evolutionary conservation of methylation reprogramming. And we established another major model in the lab, Marchantia polymorpha. And this the species has diverged from flowering plants about 450 million years ago, and importantly, has swimming sperm instead of pollen. So in a nutshell, the DNA methylation in flowering plants is targeted to transposons and the bodies of some genes. And I've just shown you that in the flowering plant germline, there is relatively subtle methylation reprogramming occurring at hundreds of genes. And in the Marchantia somatic tissues, only transposons are methylated and genes are not. However, in the Marchantia sperm, we found the whole genome is methylated. And this whole genome methylation pattern has never been seen from any plant. And in fact, as far as we know, this represents the most heavily methylated genome seen from any organism. So to understand what is driving this whole genome methylation, a postdoc, Jingyi Zhang, she obtained this map of methylome and transcriptome throughout different stages of male development. And through this, she observed that this methylation really only comes about at the very last stage of sperm maturation, when two new methyltransferases are specifically transcribed. So the methylation I've been telling you so far refers to the methylation on the 5th carbon. And there is actually another form of methyl cytosine called 4-MC with the methyl group added to the 4th amino. And 4-MC is common among bacteria and archaea, but has not been identified in eukaryotes. And the 2 new enzymes we found in Marchantia sperm have the catalytic domain of the 4-MC methyltransferases. So we performed LC-mass spec to confirm this. So here you can see that there is a nice 5-MC peak from the thallus DNA. So a thallus is a leaf-like tissue in Marchantia. And in the sperm, you can see there is another substantial peak of 4-MC in addition to the 5-MC. And in the sperm from the double knockout mutants of the methyltransferases, the 4-MC peak disappeared, demonstrating that these enzymes are really catalyzing 4-MC in the sperm. So we named these enzymes DN4MT1A and B, standing for DNA and for cytosine methyltransferase 1. So now I just want to quickly show you how this looks like in the genome. So here, this pink trace shows a 5-MC methylation detected by bisulfite sequencing. And here it shows the 4-MC, and this is detected by PacBio-SmartSeq. And because we have this developmental series, so we observe that 5-MC is actually catalyzed first. And then at the very last stage of sperm maturation, when these 2 enzymes specifically express, 4-MC is catalyzed in the genome at where there is no 5-MC. And this specific timing and scale of 4-MC methylation, that suggests that this might be important for sperm function. And indeed, we find DN4MT1 mutant sperm swim much slower and less directionally than wild type. So in short, through this study, we discovered a new functional DNA modification in eukaryotes. And this obviously opens up many exciting questions that we're pursuing further in the lab. Okay, so now let's move on to the second part regarding chromatin reprogramming. So in this theme, a process that we're very interested in is this 2 mitosis that lead to the production of sperm and vegetative cell. Because these 2 mitotic divisions give rise to drastically different chromatin states in these cells that are both haploid. So the sperm nuclei are the most compacted in a plant, whereas the vegetative nucleus is the most diffuse. And you could imagine this chromatin dimorphism is probably important for the distinctive functions of these cells. The sperm cells are relatively quiescent, and they need to pass through a long pollen tube to reach the egg cells deeply embedded in maternal tissues. Whereas the vegetative cell is the fastest growing plant cell, where there is huge evolutionary pressure to be active and get there first to reach the egg. So when a PhD student, Toby Batris, joined the lab, he performed mass spec experiments on purified sperm and vegetative nuclei. And through this, he identified a number of chromatin factors that contribute to this chromatin dimorphism. So for example, we found that the linker histone H1 that binds to the linker DNA between nucleosomes is specifically depleted in the vegetative cell. And our work demonstrated that this H1 depletion decondenses the vegetative cell chromatin and facilitates the access of a DNA demethylase called Demeter to access the DNA and demethylate. And I show this because I think this is a nice illustration of how the studies of DNA methylation and chromatin are very well interconnected. So on the sperm side, Toby discovered a histone H2B variant that is specifically expressed in the sperm. So you can see these 2 nuclei shown here. And this variant called H2B.8 has an intrinsically disordered region shown as IDR in the histone tail, and which is absent from all the canonical H2Bs. And this IDR attracted our attention because its presence is conserved among flowering plant species. And also, IDRs are frequently associated with the phase separation property of proteins. And because phase separation has recently been shown to induce nuclear condensates, we then hypothesized that H2B.8 may contribute to sperm chromatin condensation via a phase separation mechanism. So to test this hypothesis, we generated CRISPR knockout mutants of H2B.8 and examined their sperm chromatin using super resolution imaging. So before showing you the mutant, I first want to show you the wild-type sperm because there's something unusual about this chromatin. So you can see all these chromatin aggregates, and these are not seen in somatic cells. For example, this is a leaf nucleus, where you can only see this very diffused cyber green signal throughout the nucleus, except at these highly condensed foci at the nuclear periphery. So these condensed foci are the heterochromatin foci, enriched in the heterochromatic mark H3K9Me2. So histone 3, lysine 9 dimethylation. And you can see these heterochromatin foci are also present in the sperm. However, the rest of the nucleus, which corresponds to euchromatin, is still aggregated. So now in the mutant, you could see that the euchromatin condensation is reduced. This is actually hard to see from just one picture, but here are the quantifications that show that the euchromatin foci are smaller and less intense in the H2B.8 mutant. And we found the sperm nuclei are enlarged in the H2B.8 mutants. So here shows the sperm nuclear size in wild type and 2 independent knockout mutants of H2B.8. And this nuclear enlargement is indeed caused by the loss of H2B.8, as the H2B.8 GFP or a H2B.8 MYC transgene can fully complement the nuclear size phenotype. So these show that H2B.8 is required for the chromatin condensation and nuclear compaction in the sperm. So next, we wanted to see if H2B.8 is sufficient to drive chromatin and nuclear condensation. And to test this, we topically expressed H2B.8. So if you look at here, H2B.8 forms this distinctive foci in the nucleus. And excitingly, we observed similar foci in somatic cells expressing H2B.8. And further, in wild-type somatic cells, you have this very diffuse DEPI signal throughout the nucleus in the euchromatin. However, in the H2B.8 expressing cells, you have much brighter euchromatin signal that resembles the sperm. And H2B.8 also significantly reduced the somatic nuclear size. So then to test whether the phase separation property of H2B.8 is important for the condensation, we topically expressed H2B.8 without the IDR. And unlike the full-length H2B.8, this failed to induce euchromatin foci or to induce euchromatin condensation. And if you look at here, unlike the full-length H2B.8, which can effectively reduce the somatic nuclear size, the expression of H2B.8 without the IDR or a canonical H2B.2 cannot do so. So these suggest that the chromatin condensation and nuclear compaction are dependent on the phase separation ability of H2B.8. And indeed, I'm not showing you here, but we found H2B.8 undergoes phase separation in vitro and in vivo, and both requires the presence of IDR. So next, we really wanted to know what are these foci that are condensed and aggregated by H2B.8. So to do this, we performed H2B.8 chip-seq in sperm. And if you look at here, it says that H2B.8 is not deposited to heterochromatic transposons, but is abundant at euchromatic transposons. And at genes, H2B.8 is generally depleted from gene bodies. And here we group the genes based on their transcription in the sperm. With the lighter the color shows more transcription, then you would appreciate that H2beta8 is especially depleted from transcribed genes. So here it just shows how it looks like in the genome. On the top, this is H2beta8 abundance, and this green tree shows transcription, and these green boxes show genes, and orange and magenta boxes show euchromatic and heterochromatic transposon respectively. So you can see that H2beta8 is abundant at euchromatic transposons and regions that are not transcribed, but depleted from transcribed genes, and also low in heterochromatic transposons. So this means that H2beta8 actually aggregates euchromatin by bringing together transcriptionally silenced regions. So this interesting association between H2beta8 and transcription could arise via two possibilities. One is that H2beta8 simply suppresses transcription, or it could be the other way around. H2beta8 is excluded from transcribed regions. So to test these hypotheses, we first examined whether H2beta8 mutation or an H2beta8 ectopic expression would affect transcription in the sperm or seedlings, respectively. And as a result, we did not observe any significant gene misregulation. And finally, to test whether H2beta8 can inhibit transcriptional activation, we exposed the H2beta8 ectopic expressed seedlings to drought stress and examined the transcriptional response. And in short, we found that this ectopic expression of H2beta8 does not alter the transcriptome changes associated with drought stress. And here, I'm just going to show you two example genes that are known to be induced upon drought stress. So at the bottom, these are RNA-seq. So you can see these genes are induced upon drought stress in the H2beta8 ectopic expression line as they do in the wild type. So this gene is the same. And on the top, the ChIP-seq shows nicely that the H2beta8 deposited to these genes is evicted upon drought stress. So our data demonstrate that H2beta8 does not have an adverse effect on transcription. And this is very unusual because chromatin condensation is typically associated with transcriptional suppression. So our study not only addressed a long-standing question regarding how the sperm nuclei are condensed in flowering plants, but also through this revealed a novel mechanism of chromatin condensation that is compatible with transcription. So consistent with the importance of H2beta8 for chromatin condensation, H2beta8 is conserved among diverse flowering plant species, but absent from gymnosperms with uncondensed sperm, and also the non-seed plants and animals that use protamines to condense their swimming sperm. So our hypothesis, we believe that H2beta8 is an independent flowering plant invention to achieve a moderate level of sperm condensation. And the idea is that this level of condensation is sufficient for the non-motile plant sperm while being permissive for transcription. And with that, I would like to thank the great team of people I have, all of our collaborators and funding agencies. And if I'm allowed to make an advertisement, we're hiring postdocs now. If anyone's interested, feel free to directly email me. And thank you all very much for your attention. And I'm happy to take questions.